Dear colleagues, my name is Schurr and I'm heading the Institute of Plant Sciences at Forschungszentrum Jülich in Germany. I'm also a member of the PhenoTyping Excellence Cluster. Today I want to show to you some activities which we have on automated phenotyping in controlled and field conditions. I specifically want to show to you how we can use phenotyping to analyze crops during the entire life cycle. But before doing that, I would like to illustrate to you the importance of phenotyping for agriculture and plant sciences by this video, which we did in emphasis, the S3 project on plant phenotyping in Europe. It is obvious from this video how important plant-environment interactions are, and this is what plant phenotyping determines for breeding programs as well as for precision agriculture. For breeding, we need to characterize the genetic diversity of the populations which we want to start with. We also have to identify markers, genetic markers, which are needing phenotyping devices, phenotyping before in order to bring the genetic marker into life. We then have to establish breeder populations. Within the breeding populations, we are interested in the interaction within the genotype, the environment, and the management. Based on this, we select and develop varieties. Then we are confirming the trait, the heritability, and we also have to address the potential loss of diversity which has happened during the process of the development of a new variety. And this again requires characterizing the genetic diversity and the establishment of markers. And therefore we close the loop for phenotyping in breeding programs. In order to characterize these informations, we need to bring phenotyping into many different aspects of breeding. For characterizing the genetic resources, designing novel crops, develop pre-breeding processes in order to understand what the phenotypes are linked to, population analysis in large scale, breeding selection processes in the field, but also precision management later on when the crops are grown. With respect to technologies, this requires many different types of technologies. In-depth analysis, where we study how physiological traits are actually working. Population analysis, where we study from lab to field how populations are behaving and how we can select them. We then have to look into field and plot phenotyping, and in precision agriculture we actually work in the field to study how crops are working and how we can deliver, for example, nutrients or water to these growing crops in the field. In order to do that, we not only need all these different technologies, but also we need to understand the crops in their entire life cycle. Therefore, we have generated phenotyping modalities going from the seed to the entire plant. This includes seed trait analysis, sowing and germination, key process in the vegetative phase and in the generative phase, and in the end also quantifying the harvest and how the phenotypic traits which we expect are expressed in different harvesting processes. This all have to be included into databases, direct phenotyping going into the databases so that we can see how the whole process is developing, how the crop is actually developing over the entire life cycle, as well as allowing to share data between the different users of different analysis of different phenotypes. In order to do this, we have to have technologies which cover the analysis of seeds, the seedling establishment and the germination, the vegetative growth, as well as yield and harvesting products. And in this talk, I want to run you through a number of these technologies which we have developed in Jülich. Let us start with the seed, because the seed is always the starting point for the crop as well as for the phenotyping pipeline. And it's in many cases also the end, where the harvest is taken in, for example, grains, but also in fruits, because this is then the starting point for the next generation of seeds which we grow again. So why do we do seed phenotyping? We are optimizing the phenotyping pipeline with this by reducing the statistical variance in starting material, the different seed batches which we bring into the phenotyping analysis. At the same time, as I said before, the harvest in many cases is also seeds, and therefore we can characterize the outcome of the process on the crop. This also requires quantitative analysis of the seeds itself. But also seed physiology traits are very important. Seed properties itself, for example, the relationship between endosperm and the embryo, or the germination properties of the seeds and how they behave in different varieties and in different batches. Seed phenotyping is also needed to characterize germplasm. We are still waiting for analyzing a lot of different seed characteristics which are located in seed banks, where they are presently stored and waiting for discovery in the future for future development use in breeding processes. And last but not least, seed testing for seed industry is also very important, because they need to quantify and qualify the seed progeny for commercial environments. In Jülich, we have set up a number of different systems to measure seed phenotyping. One very special one is the phenoseeder. The phenoseeder combines a robotic platform with a number of different measuring systems and modules which allow seed phenotyping and handling. This allows us to analyze, for example, 2D and 3D traits like projected area, the volume, the mass, the density, the color, or 3D shapes. We can also use the system because it picks individual seeds to sort and classify seed batches. And we can also use it for planting the individual seeds into soil. This is how the system looks like. It is a robot which places the individual seeds in front of, for example, cameras, or puts it onto balances, and thereby allows to measure all the different seed parameters. We did this analysis with many different species so far, with model species like Arabidopsis, but also with seeds from commercial very important systems like maize or barley or wheat. But we can also do it with specialized systems like species, for example, species which are important for renewable energy or renewable resources like cedar, or also with specialized crops in medical and herbal plants. When we have analyzed the seeds, we can then also do a number of different things with it. First, we can recollect the seeds in one batch, and we can then look at the number of the seeds in one batch, and we can also characterize it, for example, at the end of the harvest. But we can also use the opportunity to measure individual seeds and then sort them. We can sort them by color, by weight, or any other trait which we can measure in the system. We can also store individual seeds in these kind of blocks here, where we then can pick afterwards individual seeds of specific characteristics and take them, for example, into planting. And we can also do direct planting from the robot, which allows us to do a large-scale quantitative analysis of the seeds before we put them into the soil. And as I said before, we can do also quick planting from the stored seeds of individual ones which we pick out of the information which we have there. Beyond the seed parameters, we are also often interested in the germination properties. And there are many different artificial systems where we look into seed germination in paper pouches, for example. But we have developed also a system which allows us to look into the seed germination in soil by MRI. This is a stacked system where we have a stack of number of pots above each other, and then we can study how the seeds are actually growing in soil. For example, and this is looking like this. So for here, what you see is how the root systems are developing in soil with a number of different seeds in the MRI. This can also be quantified because we can then use the analysis of the MRI, virtually pick individual plants from here, quantify them and segment them, and then get this kind of information. And here you can see different root types and how they are changing their germination and growing process in the root systems in the early days of their growth performance and their germination. Often, obviously, beyond the seeds, we are very interested in what happens in the vegetative phase and the generative phase. Therefore, we also have set up a number of systems in that domain. Very specific challenges are put to here through the phenotyping of roots. Roots are living in a very special environment, the soil, which has very special patterns, spatially and temporally. It's a chemically structured environment and has many different mechanical properties, which are also changing in different places in the soil. The intimate interaction between the root and the soil can very nicely be shown here in this micrograph from Michel some years ago, where we can show how this is actually tightly intermingled between the roots and the soil. Obviously, therefore, the artificial systems in agar or even in pots are not very likely to mimic what happens in the soil. Therefore, we need to have special devices for that. Michel in her keynote and also Malcolm in his seminar obviously showed very nice information about the root systems. Here I want to illustrate a little bit what we have done in Jülich through our unique root imaging systems, which allow deep phenotyping, like for example the PhenoPet, but also high throughput systems in the rhizotron systems which we have set up. Deep phenotyping to analyze the spatial and temporal dynamics of roots of individual species and individual specimen. We use MRI, magnetic resonance imaging on the one hand, and positron emission tomography on the other hand. We combine the two systems because they are delivering different information. MRI is used to look into the structure of root system, the root system architecture while the roots are sitting in the soil. This gives information about the structure and how it develops over time. PET is positron emission tomography. This builds on the projection of C11 with this cyclotron here as C11CO2, and then we can analyze with the positron emission tomography system where the radioactive carbon actually flows into the root and where it's deposited and where it's used. By co-registration of both the structure and the distribution of the carbon, we can get intimate information about how the carbon is flowing in specific structures and where it is deposited over time. All these tomographic systems allow us to measure the root systems in soil without taking them out and repeat measurements over and over again during the entire life cycle of the crop. This is the cyclotron, how it looks like, where we produce the 11C. This is delivered through the CO2, through photosynthesis into the plant, so a very natural approach to deliver the tracer into the plant, and then we can mimic and we can analyze quantitatively using the PhenoPet how the carbon is actually flowing in real time into the root system. This is how it looks like in the pictures then, we can see here different specimens of maize and how they are growing over time, how their root system is performing over time. So we have day 6, day 13, and day 20 after germination. And you can see how different the root systems are looking like. The gray values are MRIs and structures, and how different the carbon relations are. Here you can see very nicely the root tips and how many root tips are how many root tips the carbons deposited. And here you can see that carbon already here is moving into the roots themselves as a storage system. These kind of quantitative information are very nice, but they give us only short-term information. But therefore we can, on the one hand, look into short-term dynamics in minutes and hours, but by repeating this information, this analysis over and over again, over days, we can also determine and quantify how the root systems are developing over time. And the quantification, we can, for example, also look into the diameters of individual root systems and how they are developing over time, so that we get a full characteristic set of the root systems and how their quantitative traits are developing in the roots in the soil while they are growing. This is systems which we can only do with a few plants per day. We are increasing the number, but obviously we are interested also for breeders to provide systems which have a much higher capacity. And therefore we developed automated system for trait identification and screening, like for example our rhizotron systems. This is the rhizotron 1 system, which is working now since almost 10 years and is delivering root system analysis for 72 rhizotrons. We also have had a system in between of 250 rhizotrons, which is decommissioned now, and we are now in the final stage of commissioning the 3rd generation, where we have more than 1,000 rhizotrons to give a large-scale analysis of root systems of structure and function in the soil. This is how the data look like, roughly. This is a barley root system over time, over day 1 to day 20. And you can see how the root system is developing when it's growing along this slide, the transparent screen of the rhizotron. Obviously, we then do quantitative analysis of this and measure this, get information how the root system is actually behaving and how the different genotypes and soil conditions are changing the root structure. In a large-scale system, we have more than 1,000 rhizotrons. Here, these rhizotron systems are transported through autonomous vehicles to imaging boxes. We have 4 imaging boxes positioned at the moment, where we have different opportunities to measure different qualities of root systems, for example. This is a very large system with the 1,000 rhizotrons, which gives us a maximum of flexibility with respect to the analysis, with respect to the genotypes, and with respect to the soil systems which we are measuring. All these systems in-house are mostly looking into the single plant level and the root system architecture of individual plant. However, in the plant communities, in the field, we have plant communities which are where roots as well as shoots are interacting. Therefore, we have also set up large-scale container systems where we have separate domains from the roots from the field and put them into a deeper analysis how the root system and the shoot system are interacting. And finally, obviously, we are also interested in the field performance. This is mainly done through destructive root sampling with shuffleomics and many other systems, which give us also quantitative data on the root systems which we have in the field. All this data can be put together in models. We use for this SimRoot, a model which has been strongly developed in-house by Johannes Postma, which gives the opportunities to link many different traits which are very important for the root development, which you can see here. This allows us to develop idiotypes, how root systems should look like in certain environments. We also can integrate these models with phenotyping experiments and do kind of virtual testing of traits. And we can also use them to link lab to field opportunities. Obviously, we are not only interested in below ground, but also above ground. So therefore, we also use quantitative analysis by MRI for seeds. For example, here you can see that we can measure maize crops and how the development of individual seeds, kernels happens within the maize crop. But we also have developed portable MRI. This is systems which we can take to the field and even do imaging in the field. And we can use MRI as sensors, which allows us to measure the import of carbohydrates as well as the input of water within the same measuring system, also in the field. This is shown here, where we can see the dynamics of seed filling in a pot over time. This is on the one hand the increase in the total mass and the distribution into dry mass and water concentration, water amount, which is delivered to the pot over time. As you can see, we can see nice diurnal variations here, which the sensor is good in picking up. In the field, we are obviously interested in many different traits. Traits for roots, traits for the root structure, for the shoot structure and the canopy, traits for the water and photosynthesis, and many other traits around. Therefore, we also need specific sensors which are suitable for field measurements. There are many of them, and this is just a short overview here. At the same time, in the field, because we can't bring the plant to the measuring system, we have to position the sensors at the plant. There are many different positioning systems which are allowing us to measure in the field. We also have set up specific experiments in the field, like for example phase systems, where we expose the plants in the field to future CO2 concentrations and measure their responses in many different genotypes. And obviously, because we are not in controlled environment, environmental sensors, which are measuring atmospheric and soil parameters, are very important to analyze the environment of the crops which are in the field. In Jülich, we have a number of different systems which we can use in order to deliver, to position sensors in the field. We have flying systems like octocopters, zeppelins, and fixed-wing systems. We have autonomous systems, robots which are driving, and we also have cheap push-bike systems which are very effective in delivering information through the sensors which are mounted here to gain information for the breeders and the scientists. And then we obviously have to place on these different systems, positioning systems, the sensors. In this case, specifically sensors for photosynthesis, integrating systems, long-distance measuring systems, systems which are able to give images of properties of photosynthesis, and also hyperspectral systems which give a wider range of different information through the entire spectrum of the electromagnetic spectrum. The setups look like this. We have plot systems using different crops, like for example this one, but also crop systems which are at breeders' plots, at breeders' sites, where we then help the breeders to sort and to identify the different characteristics of the crops, of the varieties, of the genotypes in the field using our devices. As I mentioned before, we also have certain systems where we want to analyze the performance of the crops in different environments of the future. One very specific one is the breed phase. Here we expose the crops in the field to future CO2 concentrations in these rings here, where we then have a number of different genotypes which we can analyze for traits like yield, photosynthesis, water, relation, structural traits, but also insects and pests. For this we use this kind of three-dimensional, large-scale three-dimensional scanner, where many different sensors can be placed in this box here, and the positioning system is running over the breed phase systems and analyzing the performance of the crops which are growing at high CO2 in these systems. Last but not least, I want to shortly expose you to phenotyping networks. All these technologies and the analysis and the integration with breeding is requiring many different expertise. And therefore, in the past decade or so, there were many plant phenotyping networks which were established. On the one hand, the national platforms, like the German Plant Phenotyping Network or the French Phenome or the UK Plant Phenotyping Network. At the same time, we also established a number of international and regional networks. Specifically in Europe, we have developed EPN and EPN 2020, an emphasis on an international scale, global scale. There are crop-specific aspects, like, for example, the wheat initiative, or general purpose, where we bring together the phenotyping community on a global scale in IPPN. I don't have the time to go in details here, but I just want to illustrate 3 of them to you, because they have different purposes. The purpose of the European Plant Phenotyping Network projects is to provide access for users of phenotyping infrastructures on a European scale. The first one was EPN, and the second one, which is still running, is EPN 2020. This allows us to provide access to a number of different installations, more than 30 in Europe at the moment, where the Commission is paying for using these phenotyping devices and for the provision by the infrastructure owners. This gives a great opportunity to have access to many different opportunities of infrastructures. Many different experiments can be done here, and this is a real success story, how the use of installations from different countries can be shared between us. However, these projects, as you can see, have a limited lifetime, and therefore we developed in parallel the concept for a pan-European plant phenotyping research infrastructure. Emphasis, how this project is called, is an S3 project, which will develop a pan-European, interstate, international collaboration on phenotyping infrastructures. This includes a number of different infrastructure categories. We will go into controlled environments for high throughput as well as for deep phenotyping. Intensive field sites, like for example the breed phase, are used for detailed investigations of plants and canopies under well-monitored field conditions. Lean field is field sites which are providing basic equipment, where the environmental monitoring and the link to the network is a link of networks of field sites is given, which allows scientists and breeders to study very detailed and very close to their use the performance of crops in the field. Beyond these infrastructures, which are measuring plants, we're also developing models which integrate phenotyping pipelines and predictive models to use phenotypic data. And we set up a data and computational services to integrate and provide compatible information systems to provide access to the data across the different categories as well as between different infrastructure sites. At a global scale, International Plant Phenotyping Network, IPPN, is the facilitator to promote interaction between the world's phenotyping community. We integrate the regional and global activities. We enable knowledge and information expertise exchange. We address technology developments and implementation in different working groups, for example. And we also give advice to decision makers on state-of-the-art and future opportunities and limits of phenotyping setups. One very important instrument of IPPN is the International Plant Phenotyping Symposium. The next IPPS will be in Wageningen in October 2021. And we hope to see you all again there, hopefully physically and very hopefully in health and in a healthy status. Thanks very much for your attention, and I hope that I could show you a little bit how automated phenotyping in controlled and field conditions can deliver information throughout the entire life cycle of a crop.